Hey everyone, let's talk about adaptive immunity. We've already discussed innate immunity. Remember, that's all the non-specific defenses that your body uses to try to protect you from invading foreign pathogens like viruses and bacteria. And now we're going to focus on the specific ways that our body can target particular pathogens, and they are going to do that through the antigens that those pathogens possess. In other words, antigens are just pieces of those pathogens that allow our cells to recognize them. So what we're going to see with adaptive immunity is that it is connected to T and B cells. And within adaptive immunity, we are going to have two big pathways that we're going to talk about. We're going to talk about cell-mediated immunity, which is governed by T cells. And then we are going to talk about humoral immunity or antibody-mediated immunity, which is governed by B cells. And of course, the two interact, and so we'll discuss that as well. But before we even dig into that, this is a great time to just remind you that when it comes to specific immunity, there's two ways we can actually get that immunity. We can get it actively or passively. So what we're going to focus on today is active immunity. So before we get into it, let's just really quickly touch base on what passive immunity is. So passive immunity is when antibodies that target a particular pathogen are transferred from one source to another. The most common example of this that occurs naturally is from mom to baby through breastfeeding. So mom's antibodies transfer to baby through breast milk, and that's going to give baby protection to whatever antigens those antibodies are specific to. We can also actually harvest antibodies from one individual and give them to another individual. But in both of these examples, the individual did not make the antibodies, and therefore we consider that passive immunity. What we're going to talk about today is active immunity, where upon exposure to an antigen, a response is generated. And one thing that will happen is we will make antibodies in response to that antigen. Since we're making the antibodies, we consider it active. Now, that exposure to the antigen could be natural or artificial. So a natural exposure would be like you breathe in a flu virus, and all of the things that happen after that in response to that flu virus would be naturally acquiring that active immunity. But it's also possible that I could trigger your immune system by giving you a vaccine that contains a particular antigen, which will then trigger an immune response in you. So you are still doing the work of building that immunity, but the way you were exposed to it was artificial. So those kind of clarify what's happening in that chart. Important to note that adaptive immunity, whether we're, no matter what type of adaptive immunity we're talking about, it is highly specific. There is built-in versatility. We are going to have memory cells involved, which will help us respond more quickly on a second exposure. And also there is an inherent tolerance within the system that's designed to try to prevent us from developing hypersensitivities, although things can go wrong. Okay, so I mentioned two types of adaptive immunity. We sort of have two pathways of adaptive immunity. They are both going to start with the presentation of an antigen, and then that antigen presentation could lead to the cell-mediated immunity pathway or the antibody-mediated pathway. So if we go cell-mediated, that means that phagocytes have been activated by the presentation of antigen, which leads to T cell activation. And then from there, we will see activated T cells will seek out and target that particular pathogen that's associated with that antigen. Or we could have the antibody-mediated immunity pathway. In this case, the antigen presentation will ultimately lead to the activation of B cells. Those B cells will become plasma cells, and they will secrete antibodies, and those antibodies will destroy the target antigen. So regardless of pathway, the goal is to destroy the antigen. Regardless of pathway, they both depend on presentation of an antigen in order to initiate the pathway. And then we just need to learn the differences within the pathways. Remember, cell mediated is mostly T cells and antibody mediated is mostly B cells. But if you look at the diagram, you can see those up and down arrows, which indicates that there is interaction between the two pathways. And in fact, what we're going to see is that B cells are required. Let me try to reword that. B cells require activation by helper T cells in order for them to become plasma cells. So you can't have the antibody mediated pathway without the cell mediated pathway being activated. So how do we present those antigens? Well, antigens have to be presented to these particular kinds of cells, T cells and B cells. They can't just be floating around in the interstitial fluid or in the blood. These cells are not capable of recognizing antigens in that situation. Your innate defenses might be able to, but your adaptive defenses can't. Instead, those antigens must be presented on another protein, and the proteins are called major histocompatibility proteins, and there are two kinds. There's MHC1 and there's MHC2. They are essentially the same. They both do the same thing. They hold out the antigen so that our lymphocytes can see the antigen and react to it. The difference between them is which cells have which MHC protein, and then in turn, which lymphocytes can respond to those proteins. So any cell in your body, if it has a nucleus, can make MHC1 and it can present antigens on its surface using MHC1. So in other words, if any cell in your body gets infected by a virus, it can take a piece of that virus, an antigen of that virus, it can attach it to MHC1, and it can put it on its cell surface so that your lymphocytes can see it and do something about it. On the other hand, you have some cells that they don't use MHC1, they are going to use MHC2. And this is a very small subset of cells called antigen-presenting cells, or APCs. Within the APCs, that will include monocytes, which become macrophages, and even dendritic cells, which are another kind of phagocytic cell. All of those cells, plus the lymphocytes like B cells and T cells, they are all considered antigen-presenting cells, and they all use MHC2 instead of MHC1. So here's an image. This is a regular cell in your body that's been infected by a virus, and what happens is antigens from the virus are attached to MHC1 because this is a regular nucleated cell in your body. It's not an APC, it's just a regular cell. So it uses MHC1. It takes some antigen from the virus that has infected it. It attaches it to MHC1, sends it to the Golgi. The Golgi puts it in a vesicle, and the vesicle sends the whole complex up to the surface. And so now MHC1 is on the outside of the cell, holding out the antigen for any passing lymphocyte to see. So the fact that it's on MHC1 is what allows some cells to realize that this antigen is important and that they should do something about it. Now, an APC cell, these are phagocytic cells, and instead of using MHC1, they are going to use MHC2. So in this diagram, we can see our phagocytic cell, or APC, has engulfed a bacterium. It has then broken it down using a lysosome into little pieces, and then taken an antigen and bound it to MHC2 coming from the Golgi, and then via a vesicle, that MHC2 and antigen complex will be placed on the surface of the cell. And now that antigen is on display to other cells that recognize this MHC2 protein. So only some cells will pay attention to MHC1 or MHC2, but those MHCs are how other cells know that they should pay attention to that antigen. So, what do T cells do, and how do we get them to do it? So most of your T cells are just inactive. They're just kind of hanging out, not doing a whole lot. Some T cells can respond to MHC1 presented antigens, and other T cells can only respond to MHC2 antigens. The key is that all T cells have little receptors on them, and those receptors vary across the T cells, and that little receptor needs to match the antigen that's being displayed either by MHC1 or MHC2. Now, if the T cell we're talking about is a cytotoxic T cell or a regulatory T cell, they can only respond to antigens that are attached to MHC1. So whatever cell is displaying the antigen, they have to be displaying it on MHC1 in order for cytotoxic T cells or regulatory T cells to respond. Added little piece of information, all T cells have markers on them called CD markers, and the CD marker for cytotoxic T cells is CD8. Helper T cells, on the other hand, are a little bit different because they will only respond to antigens that are presented on MHC2. So in order for a helper T cell to recognize and respond to an antigen, it has to be presented on MHC2. Also, helper T cells also have a CD marker, just like cytotoxic T cells, but it's a different CD marker. It's called CD4. So sometimes in your book, they will refer to cytotoxic T cells as CD8 cells and helper T cells as CD4 cells. It's just different names, same thing. Now, if a T cell, a cytotoxic T cell, has a receptor that matches an antigen being presented on MHC1, and its little CD8 markers receive co-stimulation, that will activate the cytotoxic T cell. And what will happen as a result of that activation is that cytotoxic T cell will undergo mitosis a lot of times and produce a whole bunch more cytotoxic T cells that will go target that particular antigen. But it will also create a whole bunch of memory T cells for the future that can target that antigen. And then also it creates some regulatory T cells. And we don't focus a lot on those, but their job is to keep this whole business from getting out of control. After all, once we deal with this antigen, we want to shut this whole process down. So how do T cells, if they've been activated, how do they actually go out and destroy that target antigen? Well, they find the antigen by matching their receptor up to it, and then they use either perforin to poke holes in the membrane and destroy it, cytokines to induce apoptosis, or lymphotoxin to shut down the metabolism of the target cell. That CD8 that I mentioned is where co-stimulation takes place. So the receptor on the T cell has to match the antigen, and the CD8 on the cytotoxic T cell has to receive co-stimulation from the infected cell that's displaying the antigen on MHC1, just to confirm that the cell really is showing an antigen and really is infected, and that we should really go after it. It's just like a double check system to make sure we don't go out of control for no reason. So that's how we get MHC1 displayed antigens to activate cytotoxic T cells. But helper T cells can only be activated by antigens on MHC2 cells. And then if they get activated, they also require co-stimulation with their CD4. If they get activated, they will also undergo mitosis, and then they will produce lots more helper T cells, and then also memory helper T cells for the future. This all takes a while if we've never seen the antigen before, because first of all, you have to deal with the situation that the T cells with the matching receptor, they have to actually find and match up with this new antigen. And you have millions of T cells, so it could be a really long time before the right T cell with the right receptor finds this new antigen and this whole process starts. But if we've been exposed to it before, then we have a lot more T cells with that receptor, because that's what the memory T cells are. So the whole thing will go a whole lot faster the second time. So this is just a diagram of what I just was talking about. So here's our infected cell. It's displaying the antigen on MHC1. If we look at this little blow up here, we can see our cytotoxic T cell, or CD8 T cell, is here. The receptor on the T cell matches the antigen that's being displayed by MHC1 on our infected cell. And our CD8 proteins are sticking off right here. And our infected cell can co-stimulate using the CD8 protein. And that confirms that this is for real. And that is going to activate that T cell, and the T cell will divide to form more cytotoxic T cells, memory cytotoxic T cells, and some regulatory T cells. Those cytotoxic T cells that are made will then target and destroy the antigen or the cell that the antigen is associated with through perforin release, cytokine release, and lymphotoxin release. So that brings us to those helper T cells. How do we activate those? Well, like I said, it's pretty much the same thing. It's just that now the receptor on the helper T cell is going to recognize antigens presented on MHC2. They are also going to require that co-stimulation just to confirm that the whole situation is for real. And then we will also see the resulting mitosis from that activation that will lead to the production of more helper T cells and memory helper T cells. At the same time that that's happening, there are B cells out there with antibodies that may match up to the antigen that these T cells are also responding to. If a B cell has an antibody that matches this antigen, that is going to lead the B cell to stick to the antigen on whatever bacteria that has that antigen on its surface. So now the B cell is stuck to it, and it has a piece of that antigen, and that antigen attached to its antibody sensitizes the B cell. And it turns out that's really important because that sensitization has to happen before a helper T cell can activate the B cell to become a plasma cell. So let's look at that in pictures. So here is an antigen presenting cell. Remember, those are the ones that use MHC2 to display antigen. So this is some kind of phagocytosing cell. So it phagocytosed something and then took that antigen and put it on an MHC2 protein. So here's our antigen being displayed, and this helper T cell comes along and it has a receptor that matches that antigen. And then also you can see the CD4 protein right here, and we get co-stimulation from the APC to the helper T cell that activates the helper T cell, which causes the helper T cell to form more active helper T cells and lots of memory cells. The active ones will start releasing cytokines, and that's an important piece of this because those cytokines are going to be important in activating B cells. But in order for those B cells to be activated by those cytokines, they first have to be sensitized. So the B cells, the inactive ones, have to come across the same antigen that activated our helper cell we were just talking about. And the antigens bind to the antibodies on the B cells, which allows the B cell, which remember is a lymphocyte, so it has MHC2, so it's able to display that antigen on the MHC2, and now that B cell is sensitized, and now it can pair up with a helper T cell because they have a matching receptor to the antigen on the sensitized B cell, and that will allow for co-stimulation of the B cell by the cytokines that the active helper T cell is releasing, which means we now have an active B cell right here, and that active B cell can produce memory B cells, but it can also produce more active B cells that will mature to form plasma cells that will release antibodies, and these antibodies will go out and find that antigen and destroy it. And of course, if we already had these memory B cells up here, then we wouldn't have to go through that whole process I just talked about. Instead, these memory B cells could just be activated by cytokines from a helper T cell and start making antibodies right away, and we'd be able to fight this infection off a lot faster. And I'm going to end there because you can skip to the next video to talk about antibodies.